Good morning, good afternoon, good evening, colleagues. We'll try to start here on time. People are still joining, but we will begin with the introductory remarks. My name is Jeff Lejeune. I'm a food safety officer here at FAO, based in their headquarters in Rome. It's my distinct pleasure today to introduce our speaker, Marie Verhaegen, and the topic she will be talking about: mapping current knowledge on antimicrobial use resistance and their alternatives in plant health. As you'll note on the introductory slide, this is a combined effort within the FAO-led Renofarm initiative, the FAO Working Group on AMR, the FAO Sustainable Livestock Technical Network, and with support from the Republic of Korea through a funded project on enacting Codex Text on AMR. So before we start, I just have a few kind of administrative things to talk about. We just request that you keep your microphones on mute. It's helpful if you rename yourself, and I will do the same for myself, with your organization and the country followed by your name. Therefore, when you put questions in the chat, which we strongly encourage to do, they will be able to know who's asking the questions. When using the chat, please refrain from any non-scientific type discussions, such as advertising companies or commercial brands or specific products. As a disclaimer, Marie, who I'll introduce in one moment, although she works for FAO, we're required to say that this is her opinion and not the specific policy of the FAO itself. And then again, I did mention that the chat is available. Finally, the meeting is being recorded, so if you don't want your comments recorded, please do not put anything in the chat. All right, we will be able to share the PowerPoint and the recording on our FAO channel in the future as we move forward. So with that, I will stop sharing my screen. Oops. And introduce to you Marie Verhaegen. She is a PhD scientist who obtained her degree at the Catholic University of Louvain. And in 2023, she started working with FAO in the division of plant protection, specifically pest and pesticide management team of the Plant Production and Protection Division. She works in collaboration with the FAO Working Group and the Center for Zoonotic Diseases and AMR. Throughout her PhD, and as she continues to do, she has been working on understanding antibiotic use in plant production, their associated risks for plant resistance and plant health, and the potential for alternative measures. She worked in the Plant Bio project supported by the European Food Safety Authority as well before coming to FAO. So with that introduction, I will spend no more time of hers and allow her to dive directly into the presentation. If you have not already, please share your screen and begin, Marie. We look forward to this.
Thank you, Jeff, for the introduction. Can you all see my screen fine and you can hear me? Okay, perfect. So good afternoon, everybody. My name is Marie Verhaegen, and I've been working at FAO for almost 2 years with the Plant Production and Protection Division. But today I want to go back a little bit in time and talk about the Plant Bio project that was supported by the European Food Safety Authority, so EFSA, in 2022 and 2023, to which I had the opportunity to contribute to during the last years of my PhD. And the project aimed at mapping the current knowledge on antibiotic use resistance and their alternatives in plant health. So to start, I would like to show you this small video that is less than a minute long. The video was made in the framework of the project, and it really summarize the global objectives of the project, and it gives an overview also of this whole project. So there is no sound at the video. Well, when I tried it, the sound didn't work, but it's fine because it's not saying anything. Everything is written on the screen. I'll just change the quality. Okay. So as you maybe know, or you understood from the video, plant pathogenic bacteria, or PPB, cause devastating losses to crops worldwide. And for example, here on the slide, you have damages caused by Eohinia amylovora, which affects the pear orchards, but also Xanthomonas, which affects rice crops. And another example I put here is Pastonia solanacearum, which damaged the tomato plants. And so to control these diseases, some countries allow the use of antibiotics. And there is like a large variety of crops and foods that we consume every day that are potentially subject to this antibiotic treatment, such as rice, potato, kiwi, apples, pears, etc. The use of antibiotics in crop protection is considered low in comparison to antibiotic use in the veterinary or the human medicine field. However, there is a crucial lack of surveillance data, and globally, this potential contribution of the use of antibiotics in plants to the global antibiotic resistance issue is unknown. So there is also, over the last few years, there was a number of studies and reports that have really highlighted that the quantities effectively applied on crops are challenging to assess because of this lack of monitoring. And it has even been suggested that the use of antibiotics for plants might be more widespread than what was previously thought, and that it requires more attention. So that's the context in which the Plantibio project was initiated. It's divided into three main parts. The first one was focused on the use of antibiotics, where are they used, in which quantities, etc. The second part was focused on the risk of resistance associated with this use. What are the known resistance mechanisms? Is it possible that these resistant genes can be transferred between bacteria, etc.? And finally, the last part was about the potential alternatives to antibiotics that are available, but also being researched. So that's the three main axes of the project. There was a fourth objective described also, which was to communicate and disseminate the results of the project. And today I will mainly touch about the two first aspects of the project during this talk. To start, I want to tell you a little bit about the methodology that was applied to search the scientific literature. So first, 7 key reviews have been identified. And this will allow us to really evaluate the relevancy of our search strings, as I will explain in just a minute, because these key reviews must be retrieved with the search strings that were set up. The second step is to search for publication over scientific databases, and then the publication were selected thanks to inclusion and exclusion criteria. The reliability was assessed, and then the data can be extracted and summarized. To search the scientific literature, we use the PICO model, which helped us to write the question that would be the most relevant to answer the objective of the project. So the principle is that the question must contain different elements: the population of interest, the intervention, which is an action of interest, a comparison, and an outcome. And so this PICO conceptual model helped us to write the following question: does the use of antibiotics in plant agriculture affect the prevalence of antibiotic resistance in plant pathogenic bacteria? So this question can then be translated into different relevant keywords, which will eventually result in an appropriate number of articles when searching the different databases of scientific literature. So this question is really just an example among several questions that were defined with the model, because I cannot present all of them today. So I just want you to understand the process behind this search of literature. So here you can see the translation of the research question into keywords. And on the right, you can see the number of articles that were retrieved with each search. So it shows that it is really necessary to choose relevant keywords to decrease the number of articles retrieved in order to have relevant articles at the end. So with this method, as I said, several questions were translated into search strings. You can also see on the graph on the slide that the interest in antibiotic resistance in plant pathogenic bacteria has really increased over the last years. There are more and more publications addressing this topic. Regarding the selection of publication, there are several inclusion criteria and exclusion criteria as well. Regarding the inclusion criteria, the publication must be relevant to antibiotic application in plant agriculture. It must focus on plant pathogenic bacteria and address antibiotic resistance. And on the other hand, exclusion criteria where, so for example, indirect application of antibiotics, plant pathogenic fungi, or fungicides, etc., antibiotic resistance in other sectors. So once the articles were selected, the reliability was assessed with the help of this tool, which is called SciMago, which gives a score allowing us to appraise the quality of the journal. Okay, let's now carry on with the results from the different searches. The data the data collected were more important for streptomycin in plant agriculture. Then oxytetracycline was the second most documented, mainly as an alternative when resistance to streptomycin has been observed. Then there is a third group with casugamycin, oxolinic acid, and gentamicin. And finally, some other molecules such as nilyamycin, validamycin, etc. We see that most antibiotics used in plants are from the aminoglycosides class. And among these antibiotics, streptomycin, gentamicin, vancomycin, and validamycin were all used also in human and veterinary medicine. So only the other molecules are specific to plants. So we found about 40 countries that authorize the use of antibiotics on crops. And for this, we collected data from scientific literature, but also from the gray literature. So we looked at the official list of pesticides authorized in the country when it was available. We have established a world map that you saw in the video that I did not put again here, simply because it was made 2 years ago, and there has already been some changes in legislation in several countries, so it's not completely up to date anymore. But the map is still available in the publication that I will share the link with you later if you are curious to check it out. So we have 40 countries that authorize antibiotic use, but it does not mean that 40 countries are using antibiotics. Sometimes several antibiotics are allowed, but they are not used if there is no need, or sometimes not all antibiotics allowed are used. It also happens that some countries don't authorize antibiotic use, but there is still some unauthorized use happening because the country lacks sustainable alternatives. So it was not an easy task to list countries authorizing or not antibiotics, mainly because the data are limited. The data are limited. Their availability is limited, but also because some countries use only the commercial name of the antibiotic. That may vary in different countries, obviously. So again, this adds a difficulty to the search. I will also show you an example in the next slide, because in some countries, the data is only available in their own language. So really, this is all calling for constant data checking, and it leaves some uncertainties. There were very, very few countries which provide data on the quantity of antibiotics used. So this really remains an issue to assess the quantities of antibiotics. And as I said, really this data needs to be used with caution, because the legislation can change very rapidly, and one country can decide to forbid an antibiotic, and then it's really implemented rapidly. We still observe that the approaches followed by the countries vary greatly. Some countries completely prohibit antibiotics in plants, and the other allow the use of antibiotics. So this is the slide that I was talking about to illustrate the difficulty that was encountered during the gray literature search, notably in foreign languages. Because, for example, here in Japan, to find information, we need to look with the Japanese name of the antibiotic, which is obviously not easy when you are not a Japanese speaker. And so we have this, we had this difficulty with several countries. Also, sometimes there is not a lot of antibiotics used in plants, so the countries tend to classify the compound with the fungicide, because I mean there's not a whole category of compounds dedicated to antibiotics. So again, when we are looking for this information, it's interesting to keep in mind that sometimes we need to look for fungicides to find antibiotics. Then in this table, we have reported all the resistance cases found in scientific literature. And globally, we see that there are very few reports. So this is quite a good news. But the most represented one is streptomycin, and it was reported in 18 different countries. However, again, I want you to take this data with caution because there is a potential bias because the research is mainly centralized in countries where streptomycin is used and not in other places. I wanted here to show you one world map which is edited with the places where antibiotics are used and places with reported cases of resistance. So on the map, the countries that are colored are the ones that use the antibiotic, and the ones with the dots are the ones where resistance is reported. So on the bottom, you also see the part of the table which corresponds to the antibiotic illustrated here. And I'm really here focusing for the rest of the presentation on streptomycin because it's the most documented one. And I don't have time to present all the data for the 5 antibiotics. But basically here we see that 82% of resistance cases were reported in countries where streptomycin is used, even though we keep in mind that there is a potential bias. Because, for example, in Europe, there is no studies about this type of resistance in plant pathogenic bacteria. Okay, so you all know here, I'm sure that there are several ways that bacteria can be resistant to antibiotics. Some of these resistance are intrinsic and others can be acquired, notably through horizontal gene transfer. And then this is an interesting way of acquiring resistance because it can spread very rapidly between the bacteria at a time being. And that's also when antibiotic resistance is even more problematic because it really allows this exchange of genes to happen very rapidly. So there are 3 main mechanisms: transformation, transduction, and conjugation, which you probably know about. But what I want to show you is that there are different genes responsible for resistance to the antibiotics used for plants. And the resistance acquired by gene transfer can be easily transferred between bacteria, and they are often found on mobile genetic elements that bacteria can exchange. In that way, you see that there are multiple genes encoding for the same antibiotic, which means that bacteria, and in this case already in plant pathogenic bacteria, they already have several ways to resist to a single antibiotic. So this really illustrates the complexity of antibiotic resistance. There are 3 main groups, 3 main genes or group of genes associated with streptomycin resistance. The first one is a point mutation in the RPSL gene, which leads to a modification of the target, and then the antibiotic cannot recognize its target, so it cannot bind to the target, and it prevents its action. On the other hand, bacteria can also acquire different genes. For example, here we have AADA or STRAB genes, which they both modify the antibiotic, which inactivates its effect. And these STRAB genes are the most often encountered in plant pathogenic bacteria, and mainly on the transposon, which is called the TN5393. So here is an illustration of this TN5393 transposon and its different variants in plant pathogenic bacteria. So you can see that the transposon carries several genes represented here by the arrow. But most importantly, you can see that it carries STRA and STRB. And so these genetic structures of transposon are relatively simple. But this TN5393 is not only found in plant pathogenic bacteria, but also in human and animal pathogenic bacteria, and even in other cases in environmental bacteria. So you can see here some example with some very well-known human pathogens such as Salmonella or Klebsiella. And you see also that the variants of the transposon are way more complex in these human pathogens compared to the ones we saw on the previous slide. So for example, TN5393L has an insertion of another transposon inside the transposon itself. And the same goes for TN5393H. So really this shows that the structures of the transposons evolve over time and they can gain other features such as other mobile genetic elements, but also other antibiotic resistant genes, as for example, the transposon TC393H also has tetracycline resistant genes. So it means that when the transposon is moving from one bacteria to another, it also carries tetracycline resistant genes in addition to streptomycin resistant genes. So this here illustrates the potential complexity that transposon might in the future attain in plant pathogenic bacteria. So as I said at the beginning of the presentation, an objective of the project was to communicate and disseminate the outcomes of this project. So if you are interested in knowing more about this topic, you can check out the literature review that was published. And the project was also presented in several events. For example, the One Health conference organized by EFSA in 2022, and also the International Congress of Plant Pathology in August 2023 in Lyon, France. So as I said at the beginning, the project also examined some possible alternatives to antibiotic use in plant protection, which I will not develop here as much because it was mainly the work of another colleague of mine. So I do not have the time to go into all the details of his very impressive work. But it's still important to remember that efficient and safe alternative control measures are urgently needed to manage bacterial diseases in plant health and avoid to resort to antibiotics. And different and numerous actually innovative control measures are being explored at the moment, such as antagonistic bacteria, competitive fungi, bacteriophages, the control of the animal vectors of plant pathogenic bacteria, so they are mainly insects. And also the integrated pest management is a good strategy to put in place to reduce reliance on antibiotics. So this is the end of this first and main part of the presentation, but just to recap some key points, there is a clear lack of data. We have seen that resistance reports are, well, often coincide, I want to say, with antibiotic use, especially for streptomycin, as it is the most documented one. But there is a bias here to be cautious about, and this really calls for more systematic research. We have seen that streptomycin resistance is very prevalent, mostly with the TN53 transposon, which is still relatively simple here. However, the same transposon is found in human pathogens. It also illustrates that using antibiotics does not only select for antibiotic resistance genes, but it's also contributing to the spread and evolution of complex vectors for these resistant genes. In this plant sector, it's also essential to not forget the influence of other factors, such as irrigation water, sewage sludge, manure, and the use of copper, which could all contribute to the presence of antibiotics on plants. So there is really a need for long-term, controlled, and repeated studies on the subject. The application of antibiotics on plants could also have an impact on the whole microbiome, because beyond phytopathogenic bacteria, plants carry many other bacteria. So it could really have an impact on the whole phytobiome of the plant, and also the microbiome of the soil surrounding the plant. So now, before finishing the presentation, I want to make some links with the current work that is ongoing at FAO. So several activities have been initiated that I would like just to briefly mention here. One of the first focus of FAO's work was to create a network of experts in the field and getting involved in different working groups, really to expand the network and get to know other experts in the field. Another initiative ongoing is the writing of a technical brief on antimicrobial use in plant health and their alternatives. Another important aspect of this work is to raise awareness and communicate on the matter, which includes updating web pages of the website, participating in seminars, delivering seminars, etc. And finally, one last part of the work was to streamline data collection on antibiotics used in plants through the development of a data collection module on the use of antibiotics in a platform that was already existing at FAO, which is called Inform, that I will just explain in a minute. But because there is a major data gap in this area, this is really something to tackle as a priority. And so different initiatives have emerged to collect this data with some surveys, but also Inform, as I was mentioning. So the first surveys collecting data were launched by the International Plant Protection Convention. They distributed 2 surveys, one on antibiotics, one on fungicides. And if we look at the results from the survey on antibiotics, about among the 85 respondents, 34% confirmed the use of antibiotics in plant production. And the most frequently cited were Kesigamycin and streptomycin. They were mostly used on tomatoes, potatoes, and rice, and the estimated quantities vary greatly, from 320 kilos to 2,700 tons per year. In addition to this, FAO is working very closely with the other organizations in a number of joint initiatives. And one of them is the development of this global system integrating data collected from each organization, which the final goal is to have really an integrated surveillance and to use this One Health approach to address AMR. So the idea is to coordinate and synergize in order to divide the efforts. So in human health, the GLASS system is already existing for since a few years, and in animals, they also have the Animute system, which collects antimicrobial use data. FAO has launched its own data collection mechanism last year, which collects AMR data in animals and food, and this year, this system will expand its activities to the collection of data on antibiotic use in plant production and protection. And so this is the Inform platform, which will allow us to do this. And this data collection mechanism for antimicrobial use in plants will take the form of a questionnaire, which will be filled in by the different focal points of the country. And several questions can be answered, both on quantitative but also qualitative data. This quantitative data concern mostly the amounts of active ingredient used every year. And the qualitative data include the application practices. So, for example, spraying, injection, seed treatment, etc. The place of application, so it could be a greenhouse, a garden, a field, or other, but also the type of plant and the bacterial disease treated. So we are now coming to the end of this presentation. This was a lot of information, but I hope that it was clear. And just some key take-home messages here. Antibiotic use in plant production is lower than for human and veterinary medicine. However, it has been completely overlooked and it is under-monitored. So I feel it's taking some actions to collect data on antibiotic use in the plant sector. Finally, as a more generic conclusion, plant health is really one key component of the One Health concept, and it is essential for sustainable agriculture, food safety and security, and it should not be neglected as it was before compared to human and animal health. Thank you very much for your attention, and I'm happy to answer any questions.
Thank you very much, Mary. I'll give you a virtual applause. Everybody is liking that presentation, full of interesting and important information. We do have a couple questions, and we do have time for questions. Let's see, where should we start here? A couple of things. When you mentioned the prevalence of resistance against streptomycin in various countries, was that among plant pathogens or among plant pathogens, and how was that? How was that determined? It was just because of passive surveillance, or what? How did they mention that? They determine that.
So I guess the question is referring to the table where I was showing there are 100 cases of resistance. So the number in the table where we reported, I think it was 104 cases of resistance to streptomycin. It is, yes, to the plant pathogenic bacteria. And it's actually 104 papers in the literature that were mentioning a case of resistance in plant pathogenic bacteria. Well, some paper mention more than one case, and so in this case we counted more than once. But basically, it is the number of articles mentioning the resistance.
Number of articles. And did you have the opportunity to explore those a little bit more to see whether those were from specific food commodities? For example, were a lot of them isolated from tomatoes, and is that resistance among a specific bacterial genera and species, or did you find that in Erwinia, or did you parse that out at all to see what is going on?
So most of these articles were from the United States, where there is a lot of there is some research that is done on this topic. So it was mostly I think in California, in Michigan also.
We're talking to combat probably after fire blight or something like that.
Exactly, yes, on pears, apples, and with Erwinia amylovora. This is the main pathogen mentioned, yes.
Did you look, I know there's a couple papers out there, and we've poked around a little bit at looking at the prevalence of resistant organisms, non-plant pathogens, on foods of plant origin. Have you looked at that at all? Because if you're applying pathogens, antimicrobials to vegetables, they have a microbiome of themselves, including occasionally both environmental organisms and foodborne pathogens, and the selection could be occurring in those. So I would encourage some people to look. There are a few review papers that have looked at that. So that's one thing. A couple other things. Going back to your comment on, I had it here. Sorry. So the number of the countries so the when you mentioned the use, I think it was in the IPPC study of amount of antimicrobials used is per country, or is it? I know that in animal production they like to relate it back to like a per kilogram of food production or something. What was the basis of the amount used?
Mm-hmm. So this is indeed per country. So the countries were able to respond to the surveys and they report the data for their country. This is a difficulty with the survey because also some countries were not responding in the same unit. So this is only the data that were reliable, I want to say, that they have kept for this the presentation. But this is a difficulty, and also we do not have like a common denominator like is used in in humans and animals. But when I was talking with our IPPC colleagues, they were saying that we should report, use a denominator that would be like the surface of the field, or like in hectare or in art, depending, or in kilometers, depending on the size. But also I think that because there is so little data, it's hard to ask the country for too much information, otherwise they get overwhelmed and they are not sharing the data. But definitely it would be interesting to have like a common denominator to be able to report it to some kind of surface.
Yeah, you in your response to it, you hit on a couple of things, little data. And I'm just kind of curious as to the countries that responded to the survey. Is there anything in common with those? I mean, some of the higher income countries actually collect data, and whether they shared it or not, that would be interesting. Those that don't collect data can't share it. But is there any trends? I'm just wondering about any biases in drawing conclusions on such limited data at this point, right?
Mm-hmm.
And what concerns that you might have about that?
Mm-hmm. For sure, there is a bias. That's for sure, that when the country has less means, I mean, it's already lots of work to monitor antibiotic resistance in humans and in animals. So when we add the plant sector in addition to that, if the country doesn't have the means to do it, then it's a lot to add another type of surveillance. So for sure there is a bias. I don't have the exact list of which country responded to the survey, but it was something that was raised by our colleagues at IPPC.
Great, thanks. So I'm working my way through the questions that come through in the chat, so you don't have to look at them. There are a lot of comments about what a great job you did and an interesting presentation. I'm not trying to grow you too much. So there are a lot of those comments, and I do agree with them. That's great.
Thank you.
There's several other interesting, maybe a little more molecular biology type questions. Just wondering if any conclusions can be drawn between the genetic architecture of the transposons that are present in the plant pathogens versus those that are found in say, human pathogens. I mean, I don't know that you can really conclude any directionality of transmission, but why do you think that there's so much more diversity in the humans, and what other things may we be able to extract from that information?
Mm-hmm. Yes, so we try to compare the sequence of the transposon, but they were similar, but then there are also some differences. So it was hard to really draw a conclusion, like if they are direct paths of transmission. Yeah, we were not able to really conclude on if there was a direct transmission. And then regarding why it's more complex in humans or animals. I guess the transposons get more complex when the selection pressure is higher, and then in humans and animals there are more use of antibiotics. So I guess the transposon is more exposed to these antibiotics, and then it gets more complex and more complex over time.
Yeah, it's definitely, especially plasmids are a good reflection of One Health. I've read papers about plasmids among human nosocomial infections, and they have genes that are related to antimicrobial resistance that have been found only in plant pathogens, and genes on the same plasmid that are derived from plants, from fish pathogens, and from environmental pathogens, and from human pathogens. So once you put them in that mixing pot, they manage to hang on to them. Talking about a mixing pot, what about the role of, say, water, irrigation, rivers, transmission? Now, is that only a concern primarily for residues, because residues run off, or also potentially for resistant organisms to maybe interconnect with each other? What is your idea? Obviously, the plants aren't grown in a void; they're in the environment, and so in the bigger picture, how do you tie this in with the environmental health issues?
I think one main link with the environment is that when the antibiotics are sprayed on the plants, for sure there is some drift away or some overspray that can go maybe on the soil or on the plant that is next to this one. So I think this is one of the main ways that the antibiotic contamination can spread from the plants to the environment. But for sure, I think regarding the water, the irrigation, it's mainly the residues that can spread in that way. But I mean, in the literature that I read, if I remember correctly, the bacteria can also be transmitted in that way, also by the runoff of the water. So I think it's all interconnected, and when we use the antibiotics on the plants, the impacts can be to other places too.
Great. So obviously, I don't know if you mentioned it in your talk here, I don't think so, but you're familiar with the WHO priority list of antimicrobials. I guess they call it the MIA list now. And then also WOA has a list of priority antimicrobials for use in animal veterinary medicine. Now, those two lists are a little bit contradictory. One is the WHO, these are the ones that maybe should not be used and preserved because we need to save them. The WOA list is the opposite, saying, hey, these are the ones we really need to use because they treat the actual animal diseases. How do you think, from a One Health perspective, the use of antimicrobials in plant production, antibiotics specifically, might fit into that kind of classification?
I think for the plant sector, we really want to keep the use of antibiotics to as low as possible. So we are really at FAO trying to promote other type of strategies and alternative control measures. I think it's really important to try to make them more available and to search for funds to look for other strategies, other bacteriophages, other antagonistic bacteria, etc. because there are some alternatives available. So we should really try to use them for plants. So yeah, I think for the plant sector, the antibiotics can really be kept to a minimum and trying other strategies when they are available. That's the main problem worldwide, because, for example, in Europe, it is forbidden to use antibiotics on plants, but we have plenty of other solutions that are available and that are used, and that Europe is it's okay to put in the work and the funds to use other solutions, which is not the case everywhere in the world. So I think in comparison to humans and animals, when you have the life of a human, of an animal in hand, you cannot always be, let's try a little bit of something else, or sometimes you resort to an antibiotic and because you need it also to work fast. I think with the plants, we have alternative strategies of management, and we need to I think we need to really promote more of these alternative strategies. For some cases, at least, we could avoid the use of antibiotics.
Yeah. So in reality, it's not that much different than human health or animal health in the kind of the FAO Renofarm initiative, where the priority or the initiative is to reduce the need for antimicrobials in the first place. So practicing integrated pest management, selecting specific varietals, attention to where the crops are being planted, and irrigation, water quality, soil amendment, all those kind of things can really reduce exposure to the pest, and therefore you don't need them all in the first place. So yeah, we're coming to the end, and I'm almost out of questions here. I'll ask one more and then give people a second to type their last question in here. You mentioned developing this network of people that have interest and also have maybe some experience or expertise. How do we, or the readers or participants, find out who to go to? Contact you or contact you? Is there a community of practice available? And we didn't rehearse this beforehand, and I don't know, and I don't mean to put you on the spot, but I'm just trying to figure out. Maybe I'm just thinking, looking at the papers that you'll share with us and looking at the authors of those papers might give a good idea of who's kind of the players in the field. But do you have any other suggestions for people that are interested to find more information?
Yes, so for sure you can always contact me, and if you want to know more or if you have any question, or I can also make you in contact with other people that we now have a good connection with. But yes, for sure, looking at the authors of the papers, there are not that many reviews on the topic. So if you look for antibiotics used for plant pathogenic bacteria, you will most likely find the main reviews, and with the authors that are the main people working on this issue to contact them.
Okay. Oops. I was just trying to share my screen here with the rest of the people. Any last questions that are coming up? No? All right. Well, we want to thank you again, Marie, for the great presentation. We have this webinar series every 2 weeks. We rotate it afternoon and morning here in Rome, so we can catch people from different hemispheres and they don't have to stay up too late at night. I think you can see my screen. 2 weeks from now, we'll have a presentation from one of my colleagues here in the Division of Agri-Food Systems and Food Safety on antimicrobial residues. That's the time there. We ask also that you take time to complete the survey, the questions, the feedback, and in doing so, you can contact us. And as I mentioned before, PDFs, I believe, of the slides will be shared along with the recording of this message. So thank you for your presentation and thank all the participants for joining. We look forward to having you engage with us again in the future. So have a great day.
Thank you.